خطين
انا فاهم والله. جاب كفر البطيخ يعني.
دكتور فرانكو نص اتكلم.
اوكي كده اوكي اقول كده. we divided the glands. Those are the sons. We divided the glands into, according to branching of duct system, into, if it's one duct along branches, it's simple.
Simple.
If the duct is branching into two, the middle duct gives two branches which, into which open the secretory unit, simple branches gland. If the duct gives the branches and the branches branch again, but the branches end by the secretory unit, we call it compound gland. If the secretory unit which open into the simple duct is tubular, it's called simple tubular gland. If the secretory unit is an alveolus or grape-like, عارفين grape؟ العنب. زي العنبة أو زي الليمون أحسن. زي just like lemon. It is called simple branches alveolar. If the secretory unit is tubulo-alveolar, it is called simple tubulo-alveolar, simple branched tubulo-alveolar, compound tubulo-alveolar. So we have simple tubular, simple alveolar, simple tubulo-alveolar. Also we have simple branched tubular, simple branched alveolar, and simple branched tubulo-alveolar. What else? Compound tubular, compound alveolar, and compound tubulo-alveolar, and you have to know the site. Okay? I already mentioned before. Site. Key. Site. Now, what is about the mode of secretion of glands? How these secretory cells deliver their secretion? Get the secretion out of the secretory cells. In merocrine, the cell secretes while it is intact. How? The secretory granule together.
الحمد لله. كيف هذا الموضوع؟
Merocrine gland.
ميرو.
The cells, these are the secretory cells. This is the duct. So what is the type of this acinus, of this gland? It is simple, tubular, whatever you can say, tubular, if it's a balloon, tubular, it's alveolar, if it's elongated and narrow, it's tubular. These are the secretory cells. Here, the duct cells. Where are the secretory granules? Are the red colored dots. How they open? Secretory granules, by electron microscope, are vesicles. What do you mean by vesicles? Membrane bound, surrounded by a membrane, just like a balloon. But the balloon contains the enzymes which have to be secreted. The vesicles unite with the cell membrane and open into the surface. So, the surface is intact, saline, and the secretion went out. What we call this? Exocytosis. What we call the type of secretion of these glands? Merocrine. So merocrine glands deliver their secretion by exocytosis, and the apical cell membrane is intact. Because the membrane of vesicles fuses with the cell membrane. The secretory cells release that contains by exocytosis with no loss of the cytoplasm or cell membrane, just like salivary glands. and exocrine part of the pancreas. الحاجات اللي بتشتغل على طول. The glands which need secretion for long time. This is the first type. Second type, apocrine glands. Don't forget the type, the method and the type. Here, apocrine glands. What does we mean by apocrine glands?
Mammary?
In apocrine glands, this is the duct. Where is okay. ما اترسمتش حلوه، المفروض ابتداء من هنا الجرانيولز دي ما تبقاش موجوده. Because these are duct cells and this is the gland. So the granules have not to be here. The secretory cells accumulate the secretion in the apical part. The granules are grouped and surrounded by the apical cell membrane. The granules are grouped in the apex, and the apical part will be removed from the cell. This is the cell membrane, and these are the secretory granules delivered by the secretory cells. So, the apical part of the cell is separated with its granules, but the cells remain intact. Okay? What is the name of this type of secretion? Apo related to apex. Apocrine secretion. What is the third type? Apocrine glands, part of the apical cytoplasm of the secretory cells is released, يعني يطلع along with, together with the secretory granules. e.g. lactating mammary gland. لكن الرسمة دي سهلة، لكن بالنسبة للمامري جلاند غلط. The drawing is easy to understand, but the drawing is wrong. Why? What is the type of mammary gland? Is it simple or simple branched or a compound gland? Compound gland. Which type of compound? Tubular or alveolar or tubular alveolar? Compound alveolar gland. But this for simplification. This is the mammary gland of female. Now, holocrine gland. What does we mean by hollow? The secretory cells accumulate the secretion until a large amount is formed and then bursts off. تنفجر. Explosion of cell with the release of secretory granules, with the release of cytoplasm, with the release of nuclei to outside. What will happen? Okay, this is a secretory cell. The secretory cell will burst. This is the nucleus, this is the cytoplasm, these are the secretory granules. We have to form new cells. Instead of the lost cells, there is cell division. Cell division occurs to form new cells instead of بدلا من the lost ones. ده بيحصل فين؟ What is the example? Sebaceous gland. What is the type of sebaceous gland? We mentioned one of two types. Simple alveolar gland or simple branching alveolar gland. What its cells secrete sebum, which is an oily secretion. The cells cannot deliver the lipid out alone, but they have to burst, تنفجر, to get the secretion out. يبقى ايه محتوى السيبم? What is the contents of sebum? Secretion, together with the contents of dead cells. Holocrine glands, they secrete cells together with its accumulated secretion is released into the duct, e.g. the sebaceous glands of the skin. The sebaceous glands, as you see here, it opens into the hair follicle. But sebaceous glands, they are called tarsal glands in eyelid, open into the surface directly. Leave it to the skin. Oh, these are the three mechanisms. They are clear. Collection. What is this type? Myocline. What is this type? The escal part is lost. Allocrine. The whole cell is lost. This is much better drawing. And replaced by division of the cells. Holocrine. Now, neuroepithelium. What is neuroepithelium? Sensory epithelium. What is its function? Sensation. Reception of stimuli for special senses. What are the types of neuroepithelium, do you know? Taste buds. Where? In the tongue. The second type, olfactory epithelium. Where? In the nasal cavity. Okay? The structure of all these will be studied well with oral cavity in second year and with respiratory system. But I have to give simple idea about it. Neuroepithelium, epithelium modified to receive stimuli. The stimulus for taste is sweat, bitter, sour, salt, and so on. Of some special sensations, and in the nose, odors, الروائح. Convert them into electrical impulses carried by nerve fibers. Neuroepithelium has three main types of cells. Sensory cells for receiving the stimuli. Receptor cells. The receptor cells are supported by another cells called supporting cells. If the two types of cells are lost, there are basal cells act as stem cells. What do you mean by stem cells? Dividing. And what is the result of its division? Formation of different types of cells. It didn't give to one type. It can give rise to different types of cells. It can give rise to the sensory cells, which send the stimulus by nerve fibers to central nervous system. It can give rise to supporting cells to support the sensory cells. It can give rise to more basal cells. Sights, taste buds for taste sensation in tongue. Palate اللي هو سقف الحلق. Pharynx اللي هو البلعوم. Olfactory mucosa in nasal cavity بتبقى في الروف. It's present in the roof of nasal cavity and upper tongue cavity. Not important, but in the nose. Here is the taste bud. What is the covering epithelium of the tongue? Tell me. This is the epithelium and this is the underlying connective tissue, supporting and nourishing. What is the type of this epithelium? Stratified squamous wide, multiple layers of nuclei, basal cells open, central cells rounded, superficial cells flattened. How we know the nuclei are flattened? How the taste bud appears? Appears as an oval area. By the way, its axis يعني المحور بتاعها perpendicular to the surface. Perpendicular to the surface. These are the nuclei of cells present. Some of them supporting, some of them sensory, some of them basal. The taste bud has an opening to outside to receive the substances dissolved in mucus. To To get the test sensation, there should be a mucus to dissolve the substance to go to the test bud. If there is an odor, the odor has to go through air, dissolve in mucus, and received by olfactory epithelium. Yes. What? If it has an odor, it will dissolve and it will stimulate the receptor cell. Very small odorous substances, its particles are very, very fine. To be odor to go through the air and to go with respiration to the nose. And so usually they are not large, just like dust or carbon of smoking or something like that. Okay? These are test buds and the three types of cells. The dark ones are sensory cells, the paler ones supporting the grounding and present in between the sensory air cells for support. While the basal cells are stem. They divide to replace the other types of cells. And let me tell you one thing. Do the nerve cells are permanent?
Me?
Nerve cells are permanent?
No.
Do the nerve cells can die and replace?
No.
Only in one side. In olfactory epithelium. Because in the olfactory epithelium there are nerve cells which are destroyed and renewed from the stem cells. But here, these are the sensory are not nerve cells. The nerve fibers go to receive the sensation only. These are not nerve cells, but sensory cells. Specializations of the apical part of epithelial cells. Yeah, what do you mean by specializations of the apical part of epithelial cells? A change in the surface. Something is characteristic to some types and not present in all types. What we can find on the surface of epithelial cells? Tell me. Cilia, to move something. What else? Microvilli, to increase the surface for uptake or secretion. For absorption or secretion. Okay? This can we find on the surface. So, on the surface of epithelial cells, we may find small projections called microvilli. To increase the surface area, just like this, this is the surface area of normal epithelial cells. When we make microvilli, this is the surface area right now. We increase the surface area for uptake and secretion. What are these? The long and the tall, they are cilia. Microvilli are present in most of the cells of the body, but maybe in few numbers. But microvilli may be collected to form a brush border. Where we can find the brush border?
Small intestine.
Small intestine. What else? Kidney. Where is the kidney? Proximal convoluted tubule. Cilia. Where we can find the cilia?
Into nose.
Respiratory tract. Do we find cilia in male, in female system?
Yes.
Where?
Vagina.
Fallopian tubes and uterus. And uterus. Okay. These are the modifications on the surface. What do you want? Okay. If there are side modifications, the epithelial cells has to be connected to each other for not to separate by friction or anything else. What makes the cells connected to each other? Cell junction. Is there one type of cell junctions? Three types of cell junctions. Where you took it? Who gave it to you? Physiology. Very good. What do you mean by tight junction?
There's no space.
In the tight junction, the cell membranes fuse together, so prevent the passage of substances in between cells. Where you can find this type of junction? In intestine. Why? Anything digested had to be taken by the cells and not to pass in between. What is the second type of junction? Cap junction. What do you mean by cap junction?
فيها جاب.
The two cell membranes. This cell membrane has tubes. This cell membrane has tube. What is the nature of a tube?
فيها جاب.
Transmembrane proteins. The membrane has proteins which go up and down to the cytoplasm. 6 of them collect to make a tube in the cell membrane. In the other cell, 6 of them form a tube in its cell membrane. The two meet together. So, the opening here to the cytoplasm of this cell. The opening there is to the cytoplasm of the other cell. So, there is conduction, communication between cytoplasm of this cell and the neighboring cell. What it transmits? Signals. Where? In the heart. Because the heart, the nerve fibers in the heart are sympathetic or parasympathetic to the SCA node, but not to cardiac muscle fibers. Who carries the impulses to cardiac muscle fibers? Purkinje cardiac muscle fibers together with the gap junction between cardiac muscle cells. Difficult? Okay. Do it can transmit amino acids from one cell to cell? Yeah. Small molecules can go from this cytoplasm to that cytoplasm?
Yes.
This is the gap junction. What is the third type of junction?
Desmosome junction.
Huh?
Desmosomes.
Desmosome. Desmosome is a type of junction called adherent junction.
Mean
Adherent junction. We mean by adherent. Make cells stick together.
it can be spirit.
Stick, just like you put a paper on a surface. It stick to the surface. But the surface of this cell stick to the cell of the neighboring cell. So, it's different to separate. Give me example of site for a desmosome. Skin. Because the skin, there is always friction. If they didn't get together by desmosome, the cells will be separated. What is the common site of desmosome? In between cells of epidermis. Connective tissue or epithelium of skin? Epithelial cells in epidermis of the skin. What else? Do the basal part of the cell can go up and down, go up in folds, into the inside and come back, into the inside and come back. And these folds contain mitochondria, just like here. This is the basal membrane. It goes on, come back, goes on, come back, goes on, come back. These are basal infoldings. The cell membrane goes to the inside. What is the space of the basal infolding? Mitochondria. What we call this? Basal infolding containing mitochondria. What is the function of this?
Funk what?
Ion transport. You get the salts out together with water. Repeat again. Repeat again. This is an columnar epithelial cell or cubical epithelial cell. In the kidney, it has to get some water from the secreted urine to the body together with some salts to keep the fluid and salts of the body. These cells have to develop basal infolding containing mitochondria. This is the base of ordinary cells. But in basal infolding containing mitochondria, the cell membrane goes in, return back. Goes in, return back. When it goes in, يبقى infolding. In to the inside, folding. What is the cytoplasm of these infolding mitochondria? Why? To give energy for transport. What else? Do the cells may have arms? And these arms interdigitate? Yes. Makes lateral interdigitations to make the cells attached to each other. Clear? Or I have to repeat?
No, clear.
So, what are the modifications on the surface? Cilia. Microvilli. If the microvilli are very numerous, they form brush border. Okay? Okay. What else? No, it's working. But I left it for a long time. Come on, baby. Yes, modifications for epithelium on the apical surface, long projections for cilia, and these are movable. Small projections for microvilli, and there are numerous microvilli on the surface. It forms brush border. Basal infoldings containing mitochondria in ion transporting cells, taking water and the ions from the surface and sending it down to the blood to return to the body. Number 3, side modifications, which are cell junctions. What are the types of cell junctions? Occluders. We close the space in between the cells to prevent the passage except through cytoplasm. So the cell will allow the selected structures only to pass, or selected substances only to pass. Number 2, adherent junction. The cells will stick together. One of the types of adherent junction is called desmosome zone, but there are others. The third type is gap junction, which is a communicating junction, communicating cytoplasm of one cell to the nearing to the cytoplasm of the adjacent cell. Now, this is the basement membrane. I'm not going through now. Formed of beta lamina reticular nerva, not right now. Specializations of the epithelial cell surface are ID, apical surface modifications, microvilli cilia, lateral surface modifications, lateral interdigitations, cellular attachment sites. What are these cellular attachment sites? Occluding junctions, adherent junction, communicating junction, another name for it, nexus or gap. Leave this right now. Leave this right now. Two, more than one junction. Either all or two can connect the cells together. Yes. What we call this junctional complex. What do we mean by complex? More than one thing. So junctional complex is forming of a group of junctions to perform multiple functions. Each one performs its function. Basal surface modifications, basal membrane enfoldings, or in I-transporting cells, hemidesmosomal junction. Do you know hemidesmosome? Hemidesmosome. We mentioned desmosome. What is hemi? Half a desmosome. The desmosome connects the cell to cell. But half a desmosome connects the cells to basement membrane or basal lamina. Okay? So we have multiple types of adherent junction I am going to go through right now. This is a cell. What are these? What are these? Apical microvilli. The great number indicates what? The branch border. What is this junction? Occludence. The space in between the cells is closed. The space of the cells is closed. And the junction is at different sites to increase or to prevent the probability of passage of substance.
Tight junction. Tight.
What? Tight junction. Occludence, another name, tight junction. Okay. Wait. Number two. This is an adherent junction. The space in between the cells is widened. This is the normal space, but here it is widened. What are these? Filaments attached to cell membrane. What is this? Adherent. So this zonular adherent encircles all around the cell, as we will see in the next picture. Desmosome adherent junction, but in a spot, in a point. Number three, gap junction. The two cell membranes come very close. So this is the ordinary intercellular space. Here, the intercellular space outside of zonular adherents widened. But here, the space is closed. What are these? Basal membrane enfoldings containing mitochondria. Microvilli. I actually mentioned it, but I say few words. What are the microvilli? Small projections on the surface. What is its structure? Elevation of the cell membrane, or finger-like projection from the cell membrane. Who keeps the microvilli standing, not tilted. واقفة على حيلها. ايه اللي يخليها زي صابع كده؟ ايه اللي في صابعي؟ بول. طب ايه السبورت for microvilli? Filaments. Filaments. What we call them? Microfilaments. What is the type of microfilaments? Thin ولا thick or intermediate? Micro. Thin filaments. Thin actin filaments. Where we come from? What is the function of microvilli?
Absorption.
Increase the surface area for absorption or secretion. If they are numerous, they form brush border. Other than that, forget about it. But you can just take an idea. These are the information I want you to know. This is a picture for microvilli by electron microscope and light microscope. Here is the microvilli on the surface of epithelial cells. Here are the microvilli by electron microscope. This is the cell membrane. What is present in the middle? Microfilaments. Microfilaments. The microfilaments is attached downwards to the network of filaments called terminal web. What are the stereocilia? What do you know about stereocilia? Yes. Very good. Stereocilia are microvilli. What is the difference between stereocilia and microvilli? Stereocilia are longer. Stereocilia are branches to increase the surface area. Where we can find stereocilia? Epididymis in the rat, if you remember. Epididymis. In the epithelium lining, epididymis. Cilia. What is the function of cilia? The function of cilia is moving something on the surface.
Okay.
Whatever it is, mucus or ovum or any something else at its site. So it's movable. What is the structure of cilia? Tell me, yes. What else? Shaft, what else? Rootlets. Very good. The cilium is forming of three structures. What projects above the surface is called the shaft. What the shaft contains? Shaft, cell membrane. Surrounding structure in the middle called axoneme. The axoneme is connected in the apical cytoplasm to what is called basal body. What is the structure of basal body? The same structure of a centriole. What is the function of centriole?
Mitosis.
Cell division. Cell division. What is, how it shares in cell division?
سنتريول طلع.
Form is suspended. What, mitotic spindle. What is the structure of mitotic spindle? Microtubules. One more time. Basal body is similar to a centriole structure because when during development the ciliated cells, centriole in ciliated cells will divide multiple times, that leading to the formation of multiple basal bodies. Each basal body will form the axoneme and take with it the cell membrane up. So, in ciliated cells, how many cilia you may find in a cell?
كثير.
كتير. How many in the respiratory tract? Up to 300 per cell. So, you may find 300 basal bodies in the apical part of ciliated cell. What is the function of, what is the structure of axoneme? Microtubules. how they move. They have dynein arms, which contain ATPase, adenosine triphosphatase. If the dynein arms, which contain adenosine triphosphatase, are deficient, what will happen? Ma'sa, what will happen? No movement for cilia. What we call this? Immotile, non-motile, celiac. What is another name? Cartagner's syndrome. What are its bad effects? What will happen to the mucus not moved in the respiratory tract? Will accumulate and they make respiration difficult. And the child, since birth, will cough, trying to get mucus out. What will happen to the sperm when he married? The cilium, the flagellum of the sperm, has the structure of the cilium. So the flagellum will not move. Fertility. What does we mean by fertility? He cannot get a baby from his wife. Why? The sperm has to move from vagina to uterus to fallopian tube to fertilize the ovum. What we call this? Infertile. We call him infertile. So it's a syndrome. What do we mean by syndrome? One wrong thing, giving different manifestation. So it's one thing. What is this one thing? Immotile cilia. But in cilia in the respiratory tract, what it will do? Cannot clean the mucus up, make cough, and the mucus will collect and bacteria will grow. What the cilia make to the sperm cannot move to fertilize the ovum. And the man will be infertile. What? The absence of movement in epithelium lining fallopian tract, fallopian tube, and uterus. It's difficult for the ovum to move to the side of the uterus. So the female may be infertile too, cannot get pregnancy. Okay? What is immotile cilia syndrome? Or Cartagner syndrome? Absence of adenine arms اللي فيهم مين؟ adenosine triphosphatase enzyme, which destroys ATP to get energy for movement. No adenine arms, no ATPase, no energy, no movement, which is the function of it. Yes, you have a question.
What is the name of the syndrome?
Of?
The syndrome. Syndrome.
Syndrome, Cartagner syndrome, another name. Immotile means non-motile. Celiac syndrome. And I told you syndrome means one defect, but it gives the signs or gives the changes in different parts of the body. Disease, one thing. Syndrome, one defect leading to different signs, different manifestations, different difficulties, different effects in the body. Okay? I think I have to stop. If you have a question, tough lecture.